Okay. Now the fermentation. Now what is fermentation? Fermentation is basically done by those microorganisms which are friendly. Now this process is a metabolic process which converts the sugars to acids, gases, or alcohol. Remember the yeast? Yeast converts the sugar into alcohol. Okay, so what does yeast is doing? Yeast is performing fermentation. It is converting the sugar into alcohol. Not only alcohol, if the sugar gets converted to acids and gases, then again the process is called as fermentation. And this fermentation is used in the preparation of curd as well as in the preparation of alcohol. Lactobacillus also do fermentation to produce curd. Okay, and other bacteria also perform fermentation to get the alcohol. Now this term was first discovered by Louis Pasteur in 1857. Louis Pasteur was the only scientist who not only discovered fermentation, discovered another term, that is pasteurization. Okay, so pasteurization is a thing, like it is just you can say it's a process to sterilize, that we'll study in the next slides as well. So curd and alcohol, these are the two fermentative products that we consume. Okay, can you name some of the other fermented food items? You
Ma'am,
don't have to unmute yourself. Uh, First click on the raise hand option. Yes, and then I'll announce your name. Yes, Bala.
ma'am, bread, ma'am, from
Bread.
class. Yes, ma'am.
Okay. Anyone else? Anyone else?
Ma'am, can I repeat the question?
I'm asking that. Can you name some of the fermented food items, Akshay?
Ma'am, bread.
Except bread. Okay, because bread has already been said by this Bala. Yes, Noorjahan.
Yogurt, ma'am.
Yogurt. Good. Sri Lakshmi.
Yes, ma'am.
Can you name any of the fermented food item? Any fermented food item? Yes, Bala. Hello.
Yes, ma'am.
Yes. Do you have any other example for fermented food items?
Yes, ma'am. Wheat flour, that's
Hmm.
atta.
Uh, Charan. Charan, can you name any of the fermented food item?
Cheese.
Okay. Good. Idli is again there. Idli is one of the example for fermented food items. How many of you have heard about chana bhatura? Wake has heard, Nurjahan has heard. So the bhatura is again the fermented food item. Okay. Yes. So what is fermentation? It is a process. It is a metabolic process in which the sugar is converted into gases, alcohol, or acids. Okay. Curd and alcohol is there. One of the fermented food item. Apart from curd, you can also add idli. You can also add cheese, the yogurt. Okay. Next topic is food poisoning. Now, what is food poisoning? Whenever the microorganisms that grow on our food sometimes produce toxic substances. Okay, those toxic substances make the food poisonous, and that result in serious illness and sometimes in most severe cases, it can even cause death. Okay, that is food poisoning. Food poisoning is what it is, a disease, or you can say an illness, that is caused by the microorganisms which grow on our food, and while growing, sometimes they produce toxic substances. Okay, if the amount of those toxic substances increases, that will eventually make the food poisonous, which further results in the serious illness, and in severe cases it can cause death. Okay, is food preservation. Now, how can we preserve our food? Look, before starting the topic, or also while teaching this topic, I have told you number of times that microorganisms can grow anywhere, they can grow in any environment. Okay, and they are not uh, substantially restricted. Okay, so they can be present in air, they can be present in water, even in pure water as well. Okay, so it means that microorganisms are in our. Am I audible clearly?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes,
so
ma'am.
okay, so from which part you were not able to understand? Was the food preservation? Okay,
Ma'am, from
so
food poison.
from food poisoning. Okay, so what is food poisoning? It is an illness that is caused by the microorganisms when the count of microorganisms exceeds. So sometimes what happens that produce toxic substances, which
Ma'am, again
may
it's
impact
breaking,
food.
ma'am.
Ma'am, your voice is breaking.
Hello, hello, class.
Yes, ma'am. Hello, ma'am.
Yes, ma'am.
Yes, ma'am.
Is it clear now?
Yes, ma'am.
Yes,
Okay,
ma'am.
ma'am.
Okay, fine. So, what is food poisoning? I was trying to just explaining the food poisoning. Food poisoning is an illness that is caused by the microorganisms. Okay, so whenever the count of microorganisms exceeds, sometimes these microorganisms tends to produce toxic substances. Okay, and these toxic substances further can make the food poisonous, which eventually can cause serious illness, or in severe cases, it can even cause death to the individual that happens to consume that particular food. Okay, now if before starting this topic, or even while teaching this topic, I had told you number of times that microorganisms are present everywhere, right? So they are present even in pure water, they are present even in pure air. So how can we prevent the, you can say, the attack of that microorganism, right? How can we avoid the exceeding count of microorganisms? Because if the number microorganism will increase on the food item, that will certainly produce some toxic substances, right? And that can cause food poisoning. So how, ultimately, how we can just prevent the food poisoning? So to prevent food poisoning, there are certain chemicals, there are certain things that can prevent that. Okay, so we have different processes for that, that comes under the food preservation. Okay, so to preserve food, we can use salts, we can use sugars, the edible oils, and vinegar. These are the common chemicals that are generally used to check the growth of microorganisms. Okay, and they are called as preservatives. Fine. So they not only just they just control the growth of microorganisms. They are not stopping the growth of microorganisms. Okay, we require microorganisms for our food, fine, but we don't want them in excession, we don't want their increased count. That's why we need to check the growth of microorganisms, and here the preservatives play their role. Now we have different preservatives, we have salts, we have sugar, we have edible oils, we have vinegar as well, we have vinegar as well, right? And these are the chemical preservatives. Apart from chemical preservatives, we have different processes. We do the heat and cold treatment that can also inhibit the growth of microorganisms. Remember, the growth of microorganisms occur at certain temperature and certain pH. So when we just lower down the pH, or if we increase the pH or temperature, that can inhibit the growth of microorganisms because the optimum temperature is not there. Okay. So let's see. So we have different methods to preserve the food. The first one is preservation by oil and vinegar. You guys have seen your mothers or your grandmothers at home preparing pickles, right? So they add oil. They actually add lot of oil in the jar, and then they prepare the pickles. So that is uh, for why the oil is being added, because to preserve that pickle, oil will make a layer which will prevent the oxidation. So once the oxidation has been prevented, it can prevent the food from different microorganisms, right? So that's why this is the first method, which is the preservation by oil and vinegar. Why oil and vinegar is added? To make a layer around that food item so that oxidation should not occur. Next is storing in airtight containers. You guys have airtight jars at your home where you put some snacks, or
Yes, ma'am.
sometimes lentils have been stored in airtight containers. So this is done to prevent the direct contact of air with the food, because air has oxygen in it, and again the oxidation will occur. So the oxidation can destroy the food. That's why the food items are stored in airtight containers. Next is cold treatment. So we have refrigerators at home. Whatever we buy from the market, we just put it in the refrigerators, be it any vegetable, be it any fruit or cakes or any dairy products. We used to put everything in the refrigerators, right? Have you thought, like, have you ever wondered why this thing is done?
Ma'am, because it's
Yes.
ma'am
Yes,
because
Bala.
and because it's very cold, so the some microorganism present in there can't survive in the temperature, ma'am.
Okay,
The
good.
bacteria and fungi can't grow at a low temperature. That's why.
Mhm. To some extent you are right. Okay, so why we are giving the cold treatment? Because the lower the temperature, the lower will be the growth of the microorganisms. Okay, like there is I have told you that at around 37 degrees Celsius, which is the normal room temperature, this is the optimum temperature for the bacteria to grow, because most of the bacteria grows at this temperature. Now this refrigerator is providing below temperature than 37, right? So most of the bacteria or most of the microorganism will not grow at this temperature, at low temperature. So that's why we use to store or preserve every food item in the refrigerators. It will prevent the growth of the microorganisms. Basically, you can prevent the growth of fungi. Okay, there are various microorganisms that will uh, not grow in the refrigerators or while doing the cold treatment. Next comes the heat treatment. So whenever we buy milk, we boil them, right? So why boiling is done? Or even sometimes when you are sick, boiled water is used,
Yes, ma'am.
right? So why boiling is done? Boiling is it is just opposite to the cold treatment. Wherever in cold treatment we are providing the low temperature, in boiling or in heat treatment we are raising the temperature above that optimum temperature which is used for bacteria to grow. Okay, so even at high temperature, very high temperature, the bacteria will not grow, and even at very low temperature the bacteria will not grow. Okay, so that's why the heat treatment is given to the food items so as to prevent the growth of microorganisms. So yeah, Vivek, you were asking some question.
Yes, ma'am. Ma'am, I said that, ma'am, I didn't understand the heat treatment, so can you repeat it?
Okay, so the thing is very simple. I'm now trying to explain it in Hindi. देखो, जैसे जो रूम टेम्परेचर होता है 37 डिग्री सेल्सियस पे, ठीक है, ज्यादातर बैक्टीरिया, ज्यादातर माइक्रोब्स उसी पे ग्रो करेंगे, ठीक है। अब अगर आप 37 डिग्री से कम का टेम्परेचर यूज कर रहे हो या फिर 37 डिग्री से ज्यादा का टेम्परेचर यूज कर रहे हो, तो कुछ बैक्टीरिया ही होंगे जो ग्रो करेंगे, बट जो मेजॉरिटी है वो तो ग्रो नहीं कर पाएगी ना, ठीक है।
Yes, ma'am.
तो इसीलिए हम क्या करते हैं, कोल्ड ट्रीटमेंट और हीट ट्रीटमेंट दे रहे हैं। कोल्ड ट्रीटमेंट में हम जो है 37 डिग्री से कम का टेम्परेचर यूज करते हैं। इन कोल्ड ट्रीटमेंट वी यूज टू हैव दिस 20 डिग्री सेल्सियस और समटाइम्स रेफ्रिजरेशन लाइक फॉर 0 डिग्री सेल्सियस इज देयर। सो एट 0 डिग्री सेल्सियस एंड एट 20 डिग्री सेल्सियस वेरी few bacteria or very few microbes are there that will tend to grow, right? And in heat treatment, what we do, we just increase the temperature above 37 degrees Celsius. So even at 30, majority of the bacteria will not grow above the temperature of 37 degrees Celsius, right? So heat treatment is given for them. So जैसे आप बॉइलिंग कर रहे हैं, वाटर की बॉइलिंग करते हैं, दूध उबालते हैं, तो उसमें आप क्या कर रहे हो, टेम्परेचर को बढ़ा रहे हो, और जब 37 डिग्री से ऊपर टेम्परेचर चला जाएगा तो बहुत कम बैक्टीरिया होंगे जो उसमें ग्रो कर पाएंगे, ठीक है। तो इस तरह से आप जो बैक्टीरिया की ग्रोथ को रोक रहे हो, उसे प्रिवेंट कर रहे हो। एनी अदर क्वेश्चन विवेक?
No, ma'am. Ma'am, this much only.
Okay, fine. So yeah, this was about the cold treatment and heat treatment. Next is preservation by common salt and preservation by sugar. So sometimes fishes are there that is used to preserve by adding a layer of common salt. The salt again add this layer and it prevents the oxidation. So once the oxidation is prevented, you can preserve the food. Next is preservation by sugar. So there are very common things, or you can say the common food items that have been stored in form of candies. Okay, so mango is there. So sometimes what you do when you have to preserve, when you need to preserve mangoes, like in summers we have mangoes, so what we do, we make mango candies. Okay, we make jams, we make pickles as well, mango pickles as well. So the preparation of jams to prepare this candies from mangoes, so what we are doing, we just preserving the mango, although the process is different, the method is different. Sometimes we are making jam, so we are preserving the mango by making, converting it by sugar or by adding sugar to it. And sometimes we are making pickles, and that time what we are doing, we are preserving the mango by oil and vinegar. Now you get it? So we have 6 main different methods by which we can preserve food. So one is by preservation by oil and vinegar. Second is storing in airtight containers. Third is by using cold treatment. Fourth one is by heat treatment. Fifth, we are having this preservation by common salt, and last one is preservation by sugar. So yes, sometimes what happens that there are milk. Okay, so there is milk that does not require heat treatment. Why? Because that is pasteurized. So what is pasteurization? Pasteurization is a technique in which the milk is heated to 70 degrees Celsius. Okay, now what is the difference between heat treatment and pasteurization? In heat treatment, what you do, you go beyond 70 degrees Celsius temperature. Okay, whereas in pasteurization, you just heat up the milk to 70 degrees Celsius for only 15 seconds, and then suddenly you have to chill it out and then store. So this is pasteurization. Now when you are increasing the temperature and suddenly decreasing the temperature to zero, or like 10 to 20 degrees Celsius, it will prevent the growth of microorganisms in the milk. Also, whatever the microbes are present in the milk at that time, they will get killed. That's why the pasteurization is done. Okay, so such milk is referred to as pasteurized milk. Most of the commercial milk that you get from suppliers, that is pasteurized milk in a tetra pack or whatever, right? So that milk is pasteurized milk. Amul milk is there. Okay, so that is an example of pasteurized milk. Have you heard about this Amul milk?
Yes, ma'am.
Yes, ma'am.
Is there any other local brand of pasteurized milk that is there in your area?
Almond. Ma'am.
Oven,
Yes.
ma'am.
Ma'am,
Oh,
almond, ma'am.
okay, okay. So that is an example of pasteurized milk. Okay, fine.
Excuse me,
So,
ma'am.
yes,
Can you please repeat it, ma'am?
uh, for pasteurization, Sri Lakshmi.
Yes, ma'am.
Okay, so Sri Lakshmi, pasteurization is a preservation method, and it is for now. Just remember that it is done for preserving milk. So what we do, we heat up the milk to 70 degrees Celsius for 15 seconds. This is something different from boiling the milk. So in boiling, we go beyond 70 degrees Celsius temperature. In pasteurization, we stick up to 70 degree, like 65 to 70 degrees Celsius temperature, and we heat up the milk for 15 seconds, and then suddenly it is chilled and stored. So this pasteurization is done to prevent the growth of microorganisms. Also, whatever the microbes are present in the milk at that time, they will get killed by this process. So the milk which is prepared by this procedure, that is referred to as pasteurized milk. Okay.
Ma'am, for
Yes,
curd,
ma'am.
what they'll do, ma'am? They're keeping in fridge the curd, ma'am.
No, they don't keep in fridge. They have a very, what you say, there is a very long pipeline. Okay, which and in the pipeline the temperature is maintained. Okay, by some stats thermostat. Okay, the temperature is maintained in the pipeline. Now what they do, they just collect the milk from the milkman. Okay, the large in the large cans, and then they just transfer the whatever the milk they have collected, they just transferred to the whole container. Okay, now that container has this regulator which by which we can control the temperature. For pasteurization, what we do, we just like a regulator, we just set up the temperature to around 65 to 70 degree. Okay, now the milk which is in the large container, that starts boiling. Once it get boiled to up to 70 degrees Celsius, we just turn up this timer. We set for 15 seconds. After exact 15 seconds, the milk is then transferred throughout the pipeline, which has the lowest temperature of around 10 degree or sometimes 4 degrees there. Okay, so the temperature varies, but it should be less than 20. It should be less than 20 degree. Okay, so the temperature of the whole pipeline is then set, and we transfer the boiled milk, the heated up milk, okay, throughout the pipeline to another container where that milk is stored. So this is the whole process. The large machines are used to do this pasteurization.
Ma'am,
And
but in
if,
houses they're doing it has differently, right, ma'am?
yeah, that is different. In houses you can just simmer the milk and heat up. You just don't have to get it properly boiled. Once it start boiling, if like if you ever have boiled the milk, you might have noticed a layer starts getting formed.
Yes, ma'am.
Okay, so once the layer starts getting formed, you just have to turn off the gas and then store it in the refrigerator.
Yes, ma'am.
So this is, you can say, the pasteurization you can do at home. You don't have to just boil up the milk at full. You just have to just turn off the gas once the layer starts forming. So it means the milk has reached the temperature for 70 degrees Celsius. Okay, then just turn off the get off the milk from the gas and store it in the refrigerator. That will be pasteurization at home. Okay.
Yes, ma'am. Thank you.
Yeah. So yes, so this was the last topic for from this chapter. In the next session, we will start another chapter. In this chapter, we have discussed the microorganisms. I'm just giving the brief. Okay, we have discussed the microorganisms, their examples. We have discussed the friendly microorganisms, the medicinal use of microorganisms, the vaccines, and what is vaccination. Next was the harmful microorganisms, the microbes that cause diseases, that covers the pathogens. Okay, next we have discussed the classification of microorganisms, in which we have discussed the bacteria, the protozoa, algae, and fungi with examples. And also we have discussed the viruses, which are on the borderline of living and non-living. So we bacteria, their characteristics, probiotics, fungi, characteristics of fungi, characteristics of protozoa and viruses. Then we discuss the harmful microorganisms along with the table that includes various human diseases, the causative microorganisms, their mode of transmission, and how can we prevent these diseases. uh, then we discuss a few of the plant diseases and some animal diseases. Today we have discussed the fermentation, okay, the food poisoning, the food preservation, different methods by which we can preserve the food items, and last one was the pasteurization of milk. So yes, if anybody has any query regarding this topic, you guys can ask me. Any question throughout the topic, throughout the chapter?
Ma'am,
No, ma'am.
No? Yes?
ma'am, I have a doubt in protozoa.
You have a doubt in protozoa. So, regarding protozoa for characteristics,
Yes, ma'am, I properly didn't understand about protozoa.
you didn't understood about the protozoa. So,
Yes, ma'am.
protozoa is a type of bacteria which are unicellular. Okay? But they have flagella and sepia. So, these are thread-like structures which are present all over their body or sometimes at one end. So the examples for it includes the amoeba, the paramecium, okay, that you might have seen, the plasmodium and the euglena. For instance, you can write out the amoeba and the paramecium. These are nothing, just the single cell microbes which has flagella and cilia, which helps in their movement. Okay, so they can move. So amoeba and paramecium is the example for it.
Ma'am,
Yes.
ma'am, also virus I didn't understand.
So viruses are the microbes that are on the, they are neither living, they are not non-living. Okay. So just like bacteria, fungi, protozoa, we consider and one thing more. Yeah, algae. So bacteria, protozoa, algae, and fungi. These four we call them as living. Okay, so they are living microbes. But in case of viruses, the viruses are neither completely living nor completely non-living. Okay, so that's why virus, we call them viruses as a borderline microbe, because the viruses are non-living when they are present in the environment, but when they get contact in with a living host, be it living human being or any animal or any plant, they eventually becomes living. तो वायरसेस जो है इन्वायरमेंट में नॉन लिविंग होते हैं, ठीक है, बट अगर वो किसी लिविंग ऑर्गेनिज्म के कांटेक्ट में आ जाए, चाहे वो कोई ह्यूमन हो, एनिमल हो या प्लांट हो, वो उस वक्त लिविंग बन जाते हैं. ठीक है, पहले नॉन लिविंग है बट किसी लिविंग ऑर्गेनिज्म के कांटेक्ट में आएंगे तो वो क्या हो जाएंगे, नॉन लिविंग, लिविंग बन जाएंगे. ठीक है. Clear? So here we
Yes,
have
ma'am.
example of HIV virus, the common cold virus, and you can also write the coronavirus. So yes. Why अब ये जो चीज इनके characteristics में I have told you that the viruses are organisms that possess nucleic acid but lack the replicating machinery. Viruses जो हैं न्यूक्लिक एसिड इनके अंदर होता है. न्यूक्लिक एसिड एक ऐसा ऑर्गेनेली है जो उसकी पूरी जितनी भी एक्टिविटीज है उसको कंट्रोल करे. Right, so nucleic acid is just the cell organelle which will control all the activities of that particular virus, but there is lack of one molecule, or you can say one component, which will not allow the multiplication of this virus. Right, so that's why viruses are non-living in environment. They have nucleic acid, which is controlling all of the activities of viruses, but the molecule which will help in the multiplication of that virus cells is not there. That's why we consider viruses as non-living. Now the viruses, when they enter into the living organism, what they do, they are using the molecule which will help them in their multiplication. So once the virus start multiplicating, it will become living. ठीक है, जब वायरस किसी लिविंग ऑर्गेनिज्म के कांटेक्ट में आ जाता है तो जो भी उसका रेप्लिकेटिंग मशीनरी है लिविंग ऑर्गेनिज्म का, वो वायरस यूज करेगा. ओके, वो वायरस उसको यूज करेगा,
Yes, ma'am.
और इसी वजह से वायरस फर्दर जो है लिविंग हो जाते हैं, क्योंकि अब उनके सेल्स का नंबर जो है बढ़ने लग पड़ता है. ठीक है, सो दैट्स व्हाई viruses are non living when they are present in environment, and when they get in contact with the living organisms, they will become living.
Yes, ma'am.
Okay, so
Understood.
yes, this was the characteristics of viruses.
Ma'am,
Yes,
I have
Saksham,
doubt in food poison, ma'am.
you have doubted
Food poison.
food poisoning.
Yes, ma'am.
Okay, what's your question?
Ma'am, somebody will eat only the homemade foods, ma'am. So they are going to outside or eating something, they will get like uh they will not feel well, ma'am. Because of what?
They're not.
If they are eating
Hmm.
only the home side food and they are coming out and they want to eat outside food means it is not digested. Because of why, ma'am?
Oh, what is the you mean to say, like, what is the reason if sometimes, like, we tend to eat some street foods and it does not get digested? So the reason for this, there are number of reasons. Food poisoning is not one of the reason. The reason could be that they're using the same oil number of times. Okay, they're reusing the oil, and that oil uh, has various chemical properties. Okay, so when you are reusing the same oil, the chemical property of that oil becomes changed, which is not properly digested by our digestive system. Another factor could be the you can say the items that they are using for preparing that food, the ingredients they are using to prepare those food. uh, that could be either we can say the contaminated one. Okay, or uh, they may be not properly cleaned. Right, so these are the factors. If someone is having outside food and it is not get digested, food poisoning, the symptoms are like you may have stomach ache. Okay, you may feel nauseous. Okay, and the body suddenly starts getting weak. Okay, so the food poisoning symptoms and someone who is having the outside food is, if not getting digested, the symptoms are somewhat different.
Okay, ma'am.
In either cases, the vomits, having vomits are same.